Thank you, everyone. Today we have a webinar produced by the Pistoia Alliance Artificial Intelligence and Machine Learning Center of Excellence. And this is titled Optimizing Kinase Profiling Programs with Deep Learning. So we have three speakers today: Matt Segall, CEO of a company Optibrium, which is a Pistoia Alliance member; Samar Mahmoud, a senior scientist in Optibrium; and Fabio Broccatelli, principal scientist of Genentech, which is also a member of the Pistoia Alliance. And with this, I would like to pass the word to our respected speakers today. Samar, since you're first on the list, why don't you go first?
Hi, Vladimir. Thanks for everyone attending. So my name is Samar Mahmoud. I'm a senior scientist at Optibrium. I have a background in drug discovery and computational toxicology, and since joining Optibrium, I've been working on using different machine learning methods, and particularly Alchemite method that we are going to describe in a bit. And I'm most interested in combining deep learning methods with expert knowledge in order to aid drug discovery researchers. Yeah.
And Fabio, please.
Hi, I'm Fabio Broccatelli. I'm a principal scientist here at Genentech. I am in the DMPK department, but I'm also a joint appointment with computational chemistry, and I'm very fortunate to collaborate with many others around the company in the area of artificial intelligence and predictive modeling, but also in vitro to in vivo correlations and PBPK.
And finally, Matt.
Thank you very much for your kind invitation, Vladimir, and thank you all for joining us today. My name is Matt Segall. I'm the CEO and co-founder of Optibrium. If you go far enough into the dim and distant past, I was a theoretical physicist in computer science, but I've been working in drug discovery for over 20 years in predictive modeling and decision analysis. So thank you all again for joining us today.
And many thanks to our speakers today for taking the time to present this very curious use case. So why don't we start?
Thank you very much. So I'm just going to say a few words in terms of the overview and then hand over to Fabio, who's going to describe some of the challenges of kinase profiling in drug discovery, the specific objectives of our collaboration, and the data set which Genentech kindly provided as the basis for the work that we've done. I'll come back and say a few words about the underlying method for deep learning imputation that we've used. As Samar mentioned, this is called Alchemite. And then I'll hand over to Samar, who did a huge amount of work on this project, and will share the results of that study with you. And then we'll come back at the end to draw a few conclusions. So with that, over to you, Fabio.
Okay, thank you, Matt. So essentially, I think it's been a year and a half, couple of years ago that we first connected about this technology. And as you know, drug discovery is a data-rich environment, and we oftentimes end up having some of the in vitro data, but not all the in vitro data, and we have to make decision in hybrid of in vitro data and in silico predictions. So the technology described by Matt at the time as a data imputation technology sounded very appealing. And maybe what you will see in the next slide is that we ended up deciding we're going to use a kinase selectivity panel for this type of exploration. And the reason is that we do have quite a large data set of kinase selectivity data. We measure this routinely across all small molecule projects, obviously more so when the target of interest is a kinase, but also when it's not non-kinase. Sometimes we do end up encountering selectivity problems. So essentially, this is important because obviously if you find that your molecule is non-selective, you may also encounter down the road unexpected safety findings, and you may end up at times having more efficacy than you think you should have, and maybe failing to understand that that's also part of a side effect. So having a good understanding of the kinase selectivity profile, I think it's key for small molecule projects. And, you know, when we have to decide what is it that we're going to test for kinase selectivity, we could potentially select up to hundreds, I think 500 different assays in different formats. But generally, we basically select 10 to 15 to be screened. So it's actually very complex to select the right kinases to test, and having a model that guide us in that selection would be extremely helpful. So that is where the interest comes from. And obviously in the back of our mind, this is a first step towards potency SAR transfer learning. And, you know, wouldn't that be great if we could start exploiting data from maybe a project from 3 years ago or 5 years ago from Campbell, and transfer some of that knowledge in the current target in a fashion comparable to few shots learning, for example. Now, as always with modeling, there is a component of having accurate predictions, but you know, use a model for long enough, for sure you will break it. You will end up in a chemical space where maybe the confidence isn't great, and you know, maybe that space is a small portion of the entire chemical space. But the reality is that may be your scaffold, so you cannot get out of that space. Therefore, it becomes very important to understand when the model is confident in a prediction and when it's not. And it was great to see that with Alchemite, this was actually possible and worked out pretty nicely. So in the next slide, we're gonna briefly enumerate the collaboration objectives. Obviously, we wanna assess the ability of the method in imputation, essentially filling in missing values from these large sparse matrix. And essentially, there are two sources of information in this method potentially. One is the chemical structure of the molecule. The other one is the biological fingerprints. Maybe I have data on CDK whatever, and another CDK, for example, could be closely related in most cases, or an MPS1 or other different kinases. And essentially from that similarity in the SAR from different kinases, just from near neighbor approaches, maybe there is a lot of information that the model can already gain. So essentially, we tested both sources of information. You will see that nicely described by Samar. And we also compare this method with more traditional QSAR approaches, including deep learning. And obviously, it's valuable to have predictions, but sometimes what we want to know is what data point do I need to gather to basically enrich the information gain of my data set. So that is also something that we have looked into, and Samar has done a wonderful analysis in that area. So I think my last slide describes the data set here. We have a pretty large chemical space. It's 11,000 compounds that, importantly, they come from many different projects. So there is a lot of chemical diversity, which is nice. However, given that we're looking at hundreds of different assays, some of which are IC50s, and I would say 90% of which are percentage of inhibition data, it's really, although the data set is big, it's probably not enough to build reliable single target models for most of these kinases. Overall, close to 4% of the matrix is complete, but actually 96% of that is missing. And we are looking into completing that with in silico predictions using Alchemite. With that, I think I can pass the ball back to Matt.
Well, I think before that we have a poll, which I think Vladimir is going to run just to get some feedback from the audience.
So I have just launched polling. Please take a second to vote. And as soon as the majority of the participants had a chance to vote, I'll close the polling and I'll share the results with you. Here are the results. The overwhelming outcome is that investments in AI are high priority for our audience. So thank you very much for taking this vote. And we continue with our presentation.
There we go. So I'm just going to say a few words now just to introduce you to the method that we've actually used called Alchemite for deep learning imputation. And just as a starter, I thought I would define this term imputation and what it actually means. So I think we're all probably familiar with the idea of prediction, particularly in our field, QSAR models, that uses a set of input features to predict one or more properties for a compound. In our case, we're looking at a profile of kinase activities. So to do this, we have our features, in our case, are descriptors calculated from the molecular structure for all of the compounds in our data set. We can calculate every single value for every single descriptor, so this matrix is complete. And we can use our favorite modeling method here, for example, is random forest sort of illustrated, but it could be SVMs or Gaussian processes or deep neural networks or whatever it might be, and then make predictions for our panel of kinases or any other properties that we're interested in. Imputation is quite different. What it does is it starts with the sparse data that we have available. So here illustrated is a sort of matrix of assay data. So the little green squares represent the measurements that have actually been made. The white space, of course, are the missing values. And as Fabio described, in the data set we're working in here, 96% of that space is missing, and only 4% of those measurements have actually been made in practice. And what imputation does is to use that limited data that are available to fill in the gaps, the purple squares here on the right-hand side, using that information. The specific method that we use is called Alchemite. This is a collaboration with a company called Intelligens. They're a spin out of Cambridge University here in the UK. They have a very strong background in material science and developed this method within that domain, and have had fantastic results in, for example, designing new super alloys based on nickel super alloys for turbine blade manufacture, 3D printing materials, and so on. But we have an exclusive partnership with them to take this method and apply it in the drug discovery and small molecule optimization space. And what this does is that it takes not only the descriptors as input, again, the orange squares here, but also this sparse data that are available. And what this means is that it can learn not only from the structure-activity relationships, the relationships between the descriptors and the different assays, but also directly from the relationships between these different experimental endpoints. Even though most of that data is missing, the data that are there are enormously valuable for understanding that relationship. So it makes much better use of the sparse and, of course, noisy experimental data that we have in drug discovery than conventional QSAR models. It uses that to fill in the gaps, again, the purple squares here on the right-hand side, and as we'll also show you, can make predictions for virtual compounds. So compounds with no experimental data based purely on the descriptors of the compound structure to generate more accurate predictions and help us to target those high-quality compounds that we're interested in. The details of the method have been published in a couple of papers I've referenced here at the bottom in Journal of Chemical Information and Modeling. Another important feature relates back to Fabio's comment about the need to understand the confidence in prediction. So Alchemite doesn't just predict a single value for each experimental data point, but it actually generates a probability distribution over possible values. So sometimes you can see distributions like this on the right-hand side, which are very sort of broad and smeared out. And that's the model saying, look, the value is somewhere around here, but I'm not really sure. I haven't got sufficient data to support this with a confident prediction. Essentially, it's got a big error bar. On the other hand, in some cases, we see these very sharply peaked probability distributions, and that's where we can have confidence. It's got a small error bar on that prediction. And of course, it's the ones on the left-hand side that we want to use to make our decisions. We only want to use the best data to make decisions. And as we'll see, we've demonstrated in many cases, as we'll show you today, that this estimate of the confidence correlates very, very strongly with the observed accuracy in those predictions. Also, where we have the greatest uncertainty, like on the right-hand side, is of course where experimental data will add the greatest value. So we can also use it to suggest the experimental measurements with the most information that will help us to further improve our predictions. So we'll be talking about two different types of models, imputation and virtual. So as I mentioned, imputation models generate predictions using the sparse assay data as input, in addition to molecular descriptors to fill in the gaps, while a virtual model accepts only molecular descriptors as input. So we can make predictions for compounds that have not yet been made or tested. So to illustrate that process, of course, we have a rigorous training and test set split. So we're always evaluating against an external independent test set. So we take our data set, we split that into training set and test set. We use the descriptors and that sparse assay data in the training set to build a model. And then we test it by taking each row of the data set, and we make an imputation of this red square using the limited data as well as the compound descriptors for that compound. We make a prediction, we impute that value, and we can compare that with the independent test set value. We do that for every data point in each row to assess our ability to impute missing data. Of course, the process for a virtual model is very similar. We have our training and test set split. The main difference is here where we're actually training the model, we actually tell the model that it will receive no experimental data as input during hyperparameter optimization. And this enables us to build a model that does not use experimental data as input. So here we have the test set based purely on the descriptors. We can make predictions for all these missing values and of course compare that with the external independent test set. And finally, just to show you a couple of ways that we'll visualize the results so that help us to assess them. On the left-hand side, we have a plot here where we've basically taken all of the endpoints in the data set, the different kinase activities in this particular case. We order them from the most accurately predicted on the left to the least accurately on the right. On the y-axis, we're showing the coefficient of determination. So 1 is perfect. We get every single prediction exactly right. 0 is equivalent to essentially a random prediction, and a negative value here is actually worse than random. And so what we see is this sort of profile of activity or of accuracies across the different endpoints. And of course, the higher this is towards the top right, the better the overall accuracy across that overall set of endpoints. And just to summarize this as a single number, we take the median value to help us to compare between different methods. On the right-hand side, we're looking at the impact of those confidence estimates on the accuracy in prediction. So here for an individual endpoint, we're showing the most confidently predicted percentage of the test set along the x-axis. So starting from 100%, this is the full test set. And as we move to the left, we're focusing on subsets with greater and greater confidence as estimated by the model. On the y-axis, we're showing the root mean square error. So this is a measure of the observed accuracy of prediction. And of course, a lower root mean square error is more accurate. And here we can see a nice example of a very strong correlation between the model's estimate of its confidence and the observed accuracy of prediction. So with that background, I'm going to hand over to you, Samar, and you'll actually share the results of this project.
Thank you, Matt. Yep. So I'll be describing the results for applying the Alchemite method to predict the kinase activity profiles. But before going into sharing the results, I'm going to quickly describe the process and the data preparation that we've done. So the first step is curating and preparing the data, and I'm going to describe this in a moment. As input for all the models, we've used 330 chemical descriptors that include 30 whole molecule properties and 302 descriptors as kind of smarts. We've used several methods where we build machine learning, different machine learning and deep learning methods, models that we compared using the independent test set. So the first model that we've used is the Alchemite imputation model, and the predictions in this case are made using the compound structure and the sparse experimental data as input. The second model is the Alchemite virtual model, where the prediction here are made using only the compound structures as input. For comparison, we've used also random forest as a benchmark for a QSAR model and GenProp model, which is a multitask deep learning network. Additionally, we've also compared these models with KNN, k-nearest neighbor imputation, and virtual models. And it's worth mentioning that the KNN imputation model does use the sparse experimental data as input in addition to the compound structure and chemical descriptors. So it's a good comparison to see how it would perform against the imputation model. So the data includes measurements for kinase activity in two formats. So first is the IC50, and another format is the single point percent inhibition. And the experimental assay endpoints are annotated for the IC50 by the unique kinase name and the assay format. And for the percent inhibition, the experimental endpoints are defined by unique kinase names, the assay format, and the concentration. So we would expect to find multiple percent inhibition experimental endpoints for the same kinase, but at multiple concentrations. We've also applied some data curation where we've removed any qualified data points, which means any measurements that are annotated by greater than or less than, they were removed. We've also applied transformations to resolve skewness. So for example, for the IC50, these were converted into PIC50. And for the percent inhibition, outliers were removed, anything less than -20% or greater than 120%. And then the values were kept to be in the range between 0 and 100. We've also defined what we call measurement groups or endpoint groups. And these are groups of experimental endpoints that are taken or conducted from the same experiment or the same test. And the reason we define these as that we don't want the Alchemite imputation model to impute endpoints that are derived from the same test or the same experiment, because this is not it's not realistic in a real application. And so what we define as an endpoint group in this case is endpoints that have the same kinase or the same kinase gene or the same kinase target. So we're even more strict on defining these measurements than being conducted in the same test, but also any endpoints that are related to a single kinase target are not used to impute each other or predict one another. Then we've applied an 80-20 split by compound into training set and a test set. And what I'm going to present afterwards are the results on the 20% independent test set. So the first piece of results that I'm showing here are the comparison of the performance of the different models on the independent test set. So here we are seeing in the plot is the profile of the different models as explained earlier by Matt, where we have the for each model endpoints ordered by accuracy in the x-axis and the coefficient of determination in the y-axis. And what we see here that the Alchemite imputation model in the green line is outperforming all other models, achieving a median R² of 0.65. We also see that the models that are based on compound structure or use only chemical descriptors as input are equivalent in performance, and these are the random forest, the Alchemite virtual, and the deep learning ChemProp method. Interestingly, we also see that the KNN imputation, although it's using the experimental data as input, it's not achieving as good as the Alchemite imputation model, where the median R² is under 0.5. And the reason behind that is the Alchemite imputation model leverage best the sparse experimental data by learning the interassay correlation. So it enables us to have a great improvement in the accuracy despite having sparse experimental data as input. And in this case, we are, as Matt and Fabio mentioned earlier, that 96% of the matrix is missing. So the Alchemite imputation method is leveraging only the available 4% in the matrix to achieve this significant improvement in accuracy. We can also focus on individual type of endpoints. So here we are looking at the results on the PIC50 test set, where these plots represent scatter plots for the observed versus predicted PIC50, where we look at the imputation model in the left hand, where you see that the data points have a strong correlation between the observed and the predicted. And if we compile all the data together, we are achieving an R squared of 0.7. The Alchemite virtual model also is performing very well and achieving an R squared of 0.6. It's worth mentioning that the error bars that are associated with each data point represent the uncertainty estimated by the corresponding model. Here we are looking at the results on the percent inhibition test set. In the left plot, we are looking at the test points generated by the for the predictions generated by the imputation model, where we see that we have a strong correlation between the observed and the predicted percent inhibition, achieving an R squared of 0.8. So despite that, there are some data points that are scattered around the identity line. The majority of points are close to the identity line in this case. And similarly, the Alchemite virtual model is also predicting the percent inhibition very well, achieving an R squared, combined R squared of 0.62. So now we have seen how well the Alchemite imputation and virtual models were able to accurately predict the kinase activity profiles. What we are going to see next are the estimated uncertainties generated by the models and how we can use these uncertainties to highlight and focus on the most accurate results. So first, let's analyze the uncertainties generated for the PIC50 test by the imputation model. So on the right hand is the scatter plot that we've just seen for all the PIC50 test points, where we've previously seen that the model achieved an overall R squared of 0.71. That is reflected in the left plot for 100% of the subset of the data. The RMSC for all the predictions is over 0.5. And we can also see from the data points how different test points have variable error bars. So if we eliminated the data points with the highest uncertainty and focused on the most confident 50% of predictions, we see how the data points become closer to the identity line. And we see that the R squared in this case becomes closer to 0.9. And that is also reflected in the plot to the left, where at 50%, the most confident 50% of the data has a smaller RMSC of just over 0.3. So it dropped from over 0.5 to around 0.3. And we can even further focus on the most confident 10% of predictions, and we see that almost all data points are on the identity line. And that represents the strong correlation between the model confidence and the observed uncertainty. We can use also the uncertainty to flag outliers that is worth taking back to the lab for further investigation. So here the second example for the uncertainties generated by the virtual model, in this case for the percent inhibition. So what we are seeing in the right hand is the plot for the observed versus inhibition for all the prediction, 100% of the percent inhibition data points. And we see that we have both data points close to the identity line and few are further away. And the RMSC for all the predictions is over 15%. However, if we eliminated the uncertain predictions and focus on the most confident 50% of predictions, we see that the data points become closer to the identity line. In fact, two clusters are emerging representing non-inhibitors and potent inhibitors. And on the left plot, we see that for 50% of the most confident predictions, the RMSE drops to under 10%. We can even focus even further more on the most confident 10% of predictions, and we see that the points become even closer to the identity line, and the error drops to around 5%. So again, we see for the virtual model that we have a very strong correlation between the estimated uncertainty and the model confidence and the observed accuracy. We can also see at highest for the most confident predictions, we have very few false positives and false negatives. So before diving further into the results, I'm going to pass to Vladimir for the next poll question.
Thank you very much. And you invited to vote in the next poll. Are you already using AI in your discovery process? Overwhelming 2/3 of participants answer yes. So some form of artificial intelligence is already used in the discovery process and organizations.
Thanks, Vladimir. So, the results we've seen so far demonstrate how the Alchemite models predict the kinase activity profiles very well, especially the Alchemite imputation. We've seen how we can leverage the uncertainties generated by the models in order to focus on the most confident results. So next, what I'm going to describe is how we can use the Alchemite method to prioritize experimental efforts and select measurements to make in order to improve the predictions for the activity profiles even further. So in a conventional drug discovery setup at the initial stages of screening, it's mainly an iterative process where we run the experiments and then we select the best compounds, which we focus on, and we would also feed the data generated from experiments to build machine learning models, which will enable us to for future iterations in selecting the best candidates. However, it would also be great if we can have a database decision for which measurements to make in order to capture the most informative data. And this is something that Alchemite can do. So Alchemite can identify which measurements to make that are expected to provide us the most information to impute missing data. And we can test this by, we can make an assessment of how the Alchemite method can select the most valuable measurements from the independent test set to impute the missing values. So in order to do so, from the independent test set, we've selected 20% selection test set, what we call here selection test set, as a withhold set. And from the remaining 80%, Alchemite is used to select the optimal subsets of the remaining data at proportions of 20, 40, and 60%. And obviously, also with the 80% of the remaining test set as input to impute the selection test set. And also for comparison, the random subsets of the remaining data were also generated. So it's worth also mentioning that the 80% for both the optimal and the random selections are identical. It's also worth mentioning a caveat in this test where where the selections are made within previously measured data points. So there is no free choice. We don't. The models don't have free choice of measurements to make, and we expect the outcomes to be better if there's a free choice of these measurements. So what I'm showing here is a visualization of the first step in this selection assessment, which is splitting the independent test set into the selection test and the selection input set. So what we are seeing here is the matrix of the data where we have the compounds in rows and experimental endpoints in columns, and the red squares or the red dots represent where the data is available. So in an 80-20 stratified split, this is 20% of the test set which is withheld, and we define this as the selection test set, and the remaining 80% is the selection input set, which is the pool from where the Alchemite can select the optimal subsets. So this is the second step in the selection assessment, where we use the selection input set where for the Alchemite to generate the optimal selection at proportions of 20, 40, 60, and the full remaining 80%. And in parallel, we also generate random subsets at the same proportions. And then the third step is using these subsets as input to the Alchemite imputation model in order to impute the selection test set. So what I'm going to show next is the results of the imputation model to predict the selection test set. So what we are looking here is the performance of the imputation model at the different percentages of the selected data versus the median R-squared overall endpoints. So the optimal selection is represented in green and the random is in blue. And we can, the first observation that we can see that as we add more data as input for imputation, we expect to see an improvement in the performance. We also see that the optimal selection outperforms the random selection in each case. Obviously, at 80 and 100%, the inputs are the same. So we've seen how the optimal selection can provide an improvement in accuracy. What we can also look at the distribution of measurements, the optimal versus the random. Is there a difference in the distribution? So the bar plots here represent the frequency of endpoints to be selected for measurement. So the x-axis in each case represent the number of measurements per a single experimental endpoint. For the optimal selection in the left and the random selection in the right. And we see that for the majority of endpoints in the optimal selection, they are not required at all. So they have zero measurement. So for the optimal selection, only a small subset actually of assays that were selected as informative to predict the selection test set. Whereas if we run a random selection of measurements, we see that at least one measurement is selected per endpoint, and this way of this method of selection requires us to conduct many experiments across different or multiple endpoints that might not be necessarily required to help in the imputation. So in other words, we can from the optimal selection, we get a large reduction in the number of assays required for a higher accuracy. So this is a visualization of the selected measurements within the selection test set. So in the left, we see the 20% optimal selection, and we see that again the majority of assays are not required, and actually we see stripes of measurements for specific endpoints that are likely to be informative in order to make predictions for the selection test set. Whereas for the random selection, it's we expect to see that the measurements are following the original distributions of data available. So we can also look at the results for the 40% optimal selection, and again we see the same pattern where the majority of assays are not required for even at the level of 40% selection. And again, we see the stripes of measurements for the important endpoint. So there are different factors that determine which assay endpoints are important. So, for example, the endpoints that have the highest inter-assay correlations, and also the variability within the assay that can reflect in the level of uncertainty or the confidence of the model. But what we try to look at is how the importance of the selected endpoints is related to the sparseness or the number of measurements available in a specific endpoint. So the plot here shows the relationship between the average rank of experimental endpoints, which reflects the importance. So the most important endpoints are towards the left, and what we see that some of the most important endpoints are mostly populated, but we can also see that some of the very populated assays are not selected to be informative. What is interesting is that we also see that many important endpoints to the left corner in the plot that have little available data, which means which also reflects that if we have a free selection of measurements, you would expect that the model would gain more improvement in the accuracy if we have data for these assays. So so I've described the performance of the Alchemite modules to predict the activity profiles, and how we can use the uncertainty estimates to focus on the results, and how we can use the Alchemite imputation method to select the most important measurements. So I'll hand to you, Matt, so that you can summarize the conclusions.
Thank you very much, Samar, for a great description of some really exciting results. Just to draw a few conclusions and highlight a few points that Samar made, I think we've demonstrated that the imputation approach, this deep learning imputation approach, shows a significantly higher performance than sort of simple KNN imputation, the virtual and Alchemite virtual models, and of course, the various different types of QSAR model that we used. And this significant improvement was achieved with actually less than 4% of the possible measurements available as input. So it really demonstrates how even a very small amount of data in an imputation approach can add an enormous amount of value in terms of the accuracy of predictions that we can make. We also demonstrated a very strong correlation between the uncertainties and the observed accuracies. This means we can clearly identify the most reliable predictions with which to make decisions, but also, as Samar indicated, highlight potential outliers which might represent sort of statistical or experimental artifacts or experimental errors, which could be missed opportunities. And so we can actually identify them for retesting or confirmation of that result. And then finally, we demonstrated how we can actually then use the resulting model to choose the most valuable experiments to run, so that you can use that to more accurately impute the missing data. So by running a small subset of kinase assays, be able to impute a broad spectrum of different kinase activities with confidence. So we can reduce the experimental costs by prioritizing just that subset that's going to add the most information. So that highlights a few points here, but really should leave the last words and the conclusions to Fabio. So over to you for a second.
Okay, sure. Thanks, Matt, and thanks, Samar. Excellent presentation. Some of my points are actually a little bit redundant, so I might not stress all of them. But in essence, what do we do with this model now that we have it? You can imagine that in an ongoing project, you may have some information from kinase structural similarity and previous literature about a subset of kinases that you would select. But then you can also use the model to select the remaining 5, 6, 10 kinases. And as you get the first batch of results, you can then redo the prediction, use the imputation, and extend that to another layer of 10, 15 kinases. And you might end up identifying selectivity problems that were not trivial to identify otherwise. I think methodologically, what is really nice about this technology is that it uses the chemical information, but it also uses the existing experimental information. It is very frustrating when you're in a project and you may have some assay data that it really doesn't go in the same direction as the in silico prediction, and that's where you really lose users for in silico models. So this is a very smart technology in that regard. And obviously there is the component of accuracy, but there is also the component of basically confidence and use for essentially active learning. Now, if I have to list a number of potential avenues, and I'm actually hinting at Matt here, what would be great to have is the ability to include protein structure information as part of the training process. You can imagine that will help with dealing with a cold start problem where you have a brand new kinase of interest for which you do not have pre-existing SAR, and maybe through structural similarity, you may be able to stand up a model much earlier. And then I think similar applications can be extended to other subclasses of proteins, but also to phenotypic assays. So that's something we could definitely look into in the future. And these are my conclusions, Matt.
Thank you very much, Fabio. And I'm so glad you mentioned those, because actually, coincidentally, we are working on a project to include protein structure information with another pharma company at the moment that we hope to be able to speak about in the future. And you mentioned extending the methods to other protein classes and phenotypic assays. Well, I think it's worth mentioning that today we've been focusing on this idea of application to kinase profiles. But in fact, we have published and presented applications of this method in a very wide range of different applications. So looking at very heterogeneous data, not only biochemical, but also phenotypic assays and ADME properties. This was in collaboration with Constellation Pharmaceuticals and was published last year in Journal of Chemical Information and Modeling. And there's a webinar describing that as well. We looked at very large scale, sort of hundreds of thousands of compounds and thousands of experimental endpoints in collaboration with Takeda, which has actually just appeared in Applied AI Letters, and also a webinar that you can view here. We looked at antimalarials, this time combined with generative methods to guide the design of new antimalarials. In vivo pharmacokinetics. And our most recent webinar was actually looking at a large sort of 300,000 by compound by 500 assay global health data set in collaboration with the University of Dundee, and looked at applications to optimization of a TB project there. So lots of different ways you can apply this method across a wide range of different questions and challenges in drug discovery. Now, just before wrapping up, It's really important to acknowledge the many people who have contributed to this project at Optibrium, Ben Irwin, at Intelligence, our colleagues Tom Whitehead and Gareth Conduit, and of course the team at Genentech, including Jeff Blaney, Marcel Hopp, Paul Barosa, John Moffat, Steve McCarroll, and Katrin Hesselgren, all contributed to the ideas and the data and the analysis of this project as well. And last of all, I hope you give me just a moment to talk about how you can actually use this method if you're interested. The Alchemite method is part of our Sorella platform, which enables active learning in drug discovery. So it really automates this entire process to update the latest data from your data sources, to populate that sparse matrix we've been discussing of compounds and experimental endpoints. It applies all the sorts of transformations that Samar described, the cleaning of the data as well, and then uses Alchemite to build both imputation and virtual models. It can use the imputation model. It uses the imputation model to fill in those blanks, the purple squares again that I showed you before, to provide that complete matrix of data, each with an associated estimate of the confidence. Of course, on a global pharma data set, this can contain tens of billions of data points. So it scales to manage this massive matrix of imputed results, make it very easily searchable to find the most interesting compounds and valuable data, and provide that access seamlessly through our StarDrop platform, but also via a RESTful API. So whatever method that you use to visualize and analyze this sorts of data, you can immediately access the same models, the same imputed values, the same suggestion endpoints, and so on. And if you want to know more, we have another webinar linked here. You're very welcome to sort of see that live and in action. So without further ado, we'll stop. And I think we have sort of 5 minutes or so to answer any questions there might be. So over to you, Vladimir.
Yes, many thanks for this very interesting presentation. There is one question that came from the audience. Approximately what proportion of the tested prediction had high, medium, or low confidence? And what was the trend between inherent experimental variability of a given kinase measurement versus the mentioned proportion with varying levels of confidence?
Absolutely. I'll chip in, and then maybe Samar wants to add something. But I think it relates to this graph that we were showing you here. In this case, the imputation model for the PIC50. It depends what you mean by a high, medium, and low confidence. But what you can see here, the blue line shows the relationship between the observed accuracy and the confidence in the prediction. So if, for example, you're happy with plus or minus 0.5 log units, then over 90% of the predictions actually had sufficient confidence to achieve that level of accuracy. If, on the other hand, you want to be more stringent and use 0.3, then it's about 35%. So you can immediately sort of see this ability to sort of pick the error bars that give you the confidence that you require for the decisions that you're looking to make. The other thing I'll mention is you raised a very good point about the relationship between the confidence in the prediction and the variability in the specific endpoint you're predicting. And that's a very important, that is certainly one very important factor. Where you see an endpoint that has a lot of variability, maybe a single 0.% inhibition will have more variability than a high quality IC50, for example, you typically will see a greater uncertainty. But that's not the only factor. It also depends, of course, on the structural similarity of the compound you're making a prediction to those in the training set. So the chemical space of the model. And also because we're interpolating not only in chemical space, but in biological space, it will depend on how many supporting data points you have for other important and relevant assays. So all of these factors come together in order to essentially give you those error bars around those predictions. So I hope that helps. Samar, have I missed anything?
Yeah, I think, yeah, that's a good explanation. We've also seen a nice relationship between the uncertainties and the ranges of predictions, especially for the percent inhibition. We've seen that the most confident results fall in the ranges of either non-inhibitors or strong inhibitors. Yeah, so
That's a really good point. And when discussing it with Fabio's colleagues at Genentech, they pointed out that actually the most accurate measurements are made at the extreme, and the greatest experimental uncertainty lies in the middle. So it's actually mirroring that sort of observation in the experimental data.
Many thanks. Well, it seems that unfortunately we're out of time. So if there are still questions lingering in the minds of the audience, please feel free to send them over by email, the most interesting questions. And this concludes our presentation today. Once again, many thanks to Matt, Fabio, and Samar for doing this talk. And we will be seeing you again at the end of this month at our next webinar. Thank you very much.